Hello, friends. Welcome to Sethi Botany classes. I'm Dr. Meena Sethi. In this video, we'll study what is the biology of Agrobacterium tumefaciens, structure of Ti-PLASMID found in the bacterium Agrobacterium tumefaciens, T region of Ti-PLASMID, how Agrobacterium mediated is gene transfer is done in the higher plant. Agrobacterium tumefaciens is a bacterium which is used for production of transgenic crops. Transgenic crops are crops which have been inserted with the gene of interest for the production of herbicide tolerant, pest tolerant, drought resistant, fertile crops. So these crops have been integrated with the genome of some other bacterium or plant, and this can be done directly with the help of Agrobacterium tumefaciens. Due to this property, Agrobacterium tumefaciens is also known as nature's smallest genetic engineering. Now, this genetic engineering is the process in which we cut the DNA from other plant with the help of restriction enzyme, or from the bacterium we can cut the DNA. We can integrate the DNA and we can join the two DNAs with the help of DNA ligase, which is called as recombinant DNA. So this whole technology is also called as recombinant DNA technology or genetic engineering. So Agrobacterium tumefaciens plays an important role in recombinant DNA technology or genetic engineering. So in this video, we will discuss about Agrobacterium mediated gene transfer. First, we will study about structure of Agrobacterium tumefaciens. Now, this is a rod-shaped bacterium which is found in the soil, and this causes disease. Disease is many dicot plant, and this disease is called as crown gall disease. Now, this is the genetic material or DNA of bacteria, and this is plasmid, which is present in the bacterium. This plasmid has tDNA, so this is called as Ti-PLASMID. Now, this region, T region, is responsible for causing the disease in the plant called as crown gall. Due to this property, scientists had an idea that if this tDNA can integrate into the DNA of the host cell, this can be used for purpose of gene transfer in higher plants. Now, first we will study about structure of Ti-plasmid, which is found in the bacterium. Now, this Ti-plasmid has a point called as origin of replication. Now, this origin of replication or ORI point is responsible for its independent replication. Now, this can replicate independently, independent of the bacterial genome. Then it also has a region. This is called as T region. Now, this T region is bounded on left side as well as on the right side by a border of 25 base pairs. In addition to this, this also has an area which is called as oncogenic genes. Now, this genes is responsible for the production of hormone, phytohormone, auxin, and cytokinins. Then it also has a region for the opine synthesis. Now, what are opine? We'll discuss later on, and this region is called as conjugative transfer. Because of this region, because of conjugative transfer, this becomes compatibility with the other strains. So this can be transferred, or this can be helping in conjugation with other strains. Then this region is called as virulence region. Without virulence region, T region will not be transferred into the host plant. So virulence region is also an essential region of the Ti-plasmid. So this is the structure of Ti-plasmid. Now this is the structure of T region of the Ti-plasmid. Now this is bounded on left and on right border by 25 base pair of repeats on each side. And this area is called as oncogenic genes, which is responsible for the production of hormone, auxins, and cytokinins. So this is causing the tumorous growth. Then this area is responsible for opine synthesis, opine catabolism. Now, this region is responsible for tumorous growth in the higher plant. So scientists had an idea that if we remove the oncogenic genes, which are responsible for causing the tumorous growth, we will insert into this region our gene of interest or foreign DNA, which we want to insert into the other plant. For that transfer, even were genes they are required. So this is the structure of tDNA, which is responsible for causing the tumorous growth in the higher plants. Now, this tDNA is about 24,000 base pair long. Now, it contains genes which are forming opines and hormones like auxins and cytokinins. Now, synthesis of plant hormone, auxins and cytokinins by tDNA is responsible for uncontrollable growth in the host plant, leading to the formation of tumors, causing a disease called as crown gall tumor. Now, opines and nopeline are two types of structures present which are being formed from the opines, nopeline and octomine. Basically, what are opines? They are amino acid derivatives used by the bacterium as source of carbon and energy. Nopeline and octopine, they provide carbon and necessary energy for bacterial growth and bacterial proliferation, helping in multiplication of the bacteria. Now, wireless region is having about 24 chains in total. We will not study much detail about it, but we will just study some of the important factors like VER-A senses acetylcysteine. What is this? We'll discuss later on. VER-G is responsible for transcriptional activator of VER-box, mean formation of DNA, messenger RNA from DNA. VER-B is responsible for production of conjugational pores between plant cell and bacteria. When bacterium enters into the plant cell, then pores will be created, and VER-B region is responsible for causing that pore and helping in transfer of the bacterium into the host cell. VER-D is responsible for cleavage of supercoiled standard substrate. VER-E is responsible for gene transfer protein. VER-C will help in transfer of the tDNA into the host genome. VER-B has an ATPase activity, which will provide energy by hydrolysis of ATP into ADP and inorganic phosphates for DNA. Now, this is the biology of Agrobacterium tumefaciens. Now, we can see how tDNA is getting integrated into the host genome. Now, this is the bacterium. This is its Ti-PLASMID. In Ti-PLASMID, this is the tDNA. Now, tDNA will enter into the host cell. Now, here, this is the region where pores are being created, and VER region present in the Ti plasmid is responsible for causing this transfer of tDNA into the host genome. Now, this is the nucleus of the host genome. In the nucleus, chromosomes are found. On chromosome, DNA is found. Now, this is the host DNA. Now, this tDNA will get integrated into the host DNA. Now, when this get integrated, now here oncogenic genes, they are responsible for causing the production of hormones, auxin and cytokinins, which will cause uncontrollable growth of the cell, leading to the formation of the tumors. When this tDNA is getting separated from this Ti plasmid, this is single stranded, but when this integrates into the host DNA, this will become double stranded. Now, after integration, the genes on the tDNA code for production of cytokinin and auxin, and also release certain plant metabolites, opines and agrosinopines. Opines are amino acid derivatives, and agrosinopines are unique sugar phosphorylated derivatives. They will provide carbon and energy source for the bacteria growth and bacteria proliferation or bacteria multiplication. Now, due to this property that tDNA has been integrated into the host, now this tDNA has been separated from this Ti plasmid, get integrated into the host genome. Due to this property, this tDNA is used for gene transfer. We insert into this tDNA our gene of interest. So we will remove the oncogenic genes from the tDNA. So when we remove the oncogenic genes from tDNA, this is called as disarmed plasmid. Plasmid having tDNA without the oncogenic genes is called as disarmed plasmid. So the genes for phytohormone and opine, they are removed from the tDNA. Only border regions and one region on the plasmid, they remain intact. Other genes have been removed. And when we remove the oncogenic genes and opine region from the tDNA, then the plasmid having such type of tDNA is called as disarmed plasmid. Now this plasmid, which is called as disarmed because it doesn't have any oncogenic region, doesn't have opine catabolism region, so this will not cause disease in the plant. But if we will insert our gene of interest into that DNA, that will get integrated into the host genome. So gene of interest or foreign DNA is inserted into tDNA of disarmed plasmid. The cells with having our gene of interest or foreign DNA will be called as transformed cells because they are having other gene from other genome. Now they can be used for the production of the transgenic crops. Now, this is how gene of interest is added. Now, this is the left border of the tDNA. This is the right border. And in this, we have added foreign DNA, or we can say our gene of interest or scientist gene of interest, which we want to incorporate into the host genome, which we want to transfer into the host genome. So that gene of interest is added here. And for selection or for screening of the transformed cell, a marker gene is added. This can be antibiotic resistant gene or other kind of marker gene can be used. Now, Agrobacterium mediated transfer DNA is widely used as a tool in biotechnology. This is being used for the production of transgenic crops. This we have already discussed because transgenic crops are the crops which have other gene of interest or foreign DNA. So, in genetic engineering, the tumor promoting and opine synthesis gene are removed from the tDNA. They are replaced with a gene of interest or a selection marker, which will help in selection of the or screening of the transformed cells. So, Agrobacterium tumefaciens is used for the production of transgenic crops in three steps. First step is production of the disease into the host plant, induction of crown gall disease, and assaying for opine synthesis. But for production of transgenic crops without disease, we will remove the oncogenic genes and opine genes. So this is called as production of the disarmed plasmid. So second step is transformation of the plant cells with disarmed plasmid, and next is selection of the transformed cells. Now, how we will come to know that these bacterium have been used for the transgenic crops and which cells of the host genome have been transformed and have gene of interest, that we can go for Gus gene assay. So Gus gene assay is used for screening and selection of the transformed cell. So Gus gene assay we have already discussed in one video that I will add a link into the description box. So in Gus gene assay, basically the cells which are transformed, they will turn blue in color, and we will pick up those blue color and used for other purposes in the genetic engineering. So in that way, we can raise the transgenic crops from the transformed cells. So first step is induction of crown gall disease and assaying for opine synthesis. Second is transformation of the plant cells with disarmed plasmid and selection of the transformed cell. Third is we can go for marker genes. For that, we can also have Gus gene assay, the use of visual assay for screening transformed cells, for example, Gus gene assay. And we can also select transformed cells, and we can use those transformed cells for production of transgenic crops. Thank you for watching the video. If you have liked the video, please like, share, and subscribe.